Welcome to Cancers 101, where we break down complex cancer biology in a way that's easy to understand. I'm Rittika, your host, and today we are discussing a crucial aspect of cancer: how cancer cells manage to bypass the cellular breaks and normal control growth. We'll take a closer look at mutations in tumor suppressor genes like P53 and RB, how these allow cancer cells to ignore growth inhibitory signals. As always, I'm joined by Aditi, an expert in cancer research. Hi, Aditi.
Hi Ritika, I'm excited to dive into today's topic. Cancer cells are notorious for ignoring the brakes that keep our cells from growing uncontrollably. So, understanding how they do that is key to developing better treatments.
To set the stage, let's imagine the normal process of cell division as a car trying to drive down the road. We have got the gas pedal, which signals the car to move forward, but we also need the brakes to make sure the car doesn't speed out of control. In a healthy cell, growth is tightly regulated. Signals tell the cell when to grow and when to stop. In cancer, these brakes often malfunction, allowing the cells to keep dividing unchecked.
Exactly. One of the main brakes in our cells is the tumor suppressor genes, genes that produce proteins responsible for halting cell division when something goes wrong. Two well-known examples are P53 and the retinoblastoma protein or RB. These proteins act like checkpoints, stopping the cell cycle when there is DNA damage or other issues that could lead to cancer. But in many cancers, mutations in these genes mean the brakes are either not working or entirely bypassed.
That's where the trouble starts. P53, for instance, is often referred to as the guardian of the genome because it senses when the DNA is damaged and can either repair it or trigger cell death if the damage is too severe. But in many cases, mutations in P53 prevent it from doing its job, allowing the cells to continue dividing despite the damage.
And it's not just P53. The RB protein plays a similar role. RB is involved in regulating the cell cycle, essentially deciding when the cell should move on to the next stage of the division. When RB is mutated or inactivated, the cell can skip over these checkpoints and continue dividing without resistance. This is particularly common in cancers like retinoblastoma and certain type of lung cancers.
So essentially, these mutations act like broken brakes in a car, allowing cancer cells to ignore the normal signals that should stop them from growing. But it's not just genetic mutations at play here. There's also epigenetic components, changes in how genes are regulated that don't involve changes to the DNA sequences itself.
That's right. In cancer, epigenetic changes can silence tumor suppressor genes without actually mutating them. For example, tumor suppressor genes like P53 and RB can be turned off by changes in DNA methylation or histone modification. Basically, molecular tags that control whether a gene is active or silent. When these genes are epigenetically silenced, the cell loses its ability to stop dividing, even if the gene itself isn't mutated.
These epigenetic signaling or silencing is a part of the cancer puzzle. It's like locking the brakes in the place so they cannot ever be used, even if the parts are still there. This interplay between genetic mutations and epigenetic changes mean that the cancer cells have multiple ways of bypassing growth inhibitory signals and continuing their unchecked proliferation.
So now we understand how cancer cells evade these critical growth suppressors. But what can we do about it? Fortunately, there are a few therapeutic strategies that aim to restore tumor suppressor function, one of the most exciting areas of research in gene therapy.
Gene therapy works by introducing healthy copies of tumor suppressor genes like P53 into cancer cells, essentially reactivating the brakes. The hope is that by restoring the function of these proteins, the cells will be forced to stop grow or even die off if the damage is too severe. While this is largely experimental, it's a promising area of research.
And on the epigenetic side, there are drugs that can reverse the epigenetic silencing of tumor suppressor genes. These drugs, known as epigenetic modifiers, can unmute the tumor suppressor genes, allowing them to function properly again. For example, drugs that target DNA methylation or histone deacetylases are being tested in clinical trials for their ability to restore the activity of genes like P53 and RB.
Let's look at a case study to see how this works in practice. Meet Sarah, a patient with small lung cancer, which is often associated with mutation in P53. Sarah was a part of clinical trial testing a gene therapy approach that aimed to restore P53 function. After receiving the therapy, her tumor growth slowed significantly and the reactivation of P53 protein began to restore the cell's ability to stop dividing. This is a great example of how restoring tumor suppressor function can have a real impact on cancer progression.
It's still early days, but studies like Sarah's show us that reactivating tumor suppressor genes could be a key strategy in cancer treatment. With continued research, we might one day be able to offer patient therapies that can bypass the need for traditional chemotherapy altogether, focusing instead on restoring the body's natural cancer fighting mechanisms.
To sum up today's episode, cancer cells can evade growth suppressors like P53 and RB, either through mutations or epigenetic silencing, allowing them to bypass the normal growth breaks. But by targeting these processes with therapies like gene therapy and epigenetic drugs, we have the potential to restore these critical growth inhibitory signals and slow or stop cancer growth.
It's exciting to think that with new advances, we may one day be able to unlock these tumor suppressor genes and restore normal cellular function, potentially revolutionizing cancer treatment.
That's all for today's episode of Cancer 101. Thanks to Aditi for sharing her expertise on cancer evades growth suppressors. If you have any questions or thoughts, feel free to reach out to us.
Thanks for having me, Ritika. It's always great to discuss how we can outsmart cancer strategies. See you next time.
Take care, everyone, and remember cancer cells can bypass the brakes, but we are working hard to find ways to hit the stop button. See you soon.